April 2011. Aisha will be talking about the two best results. Polycaria undulata, that's the yellow flower in the corner with a bee on it. And Peronychia argyrocoma, that's the white flower in the middle of the slide. This Google Earth slide will show where we collected most of our desert plants. One of the areas we collected was along the road to Doha. However, this is a major highway and often more difficult to see the plants off in the distance from the highway. The second area we collected was near Fort Zubara. This area was easier to collect because along this two-way road there were ditches that collected water, which made it easier to see the many plants that were close to the road. Also, there is much less traffic here. Ali Sharif is a local artist and a desert plant specialist who helped us identify the plants. Here is Ahmed and Aisha with Ali Sharif using a dichotomous key to identify one of the plants we collected. Here we have Ahmed and Aisha again collecting Cistantia tubulosa. On the next slide, I'm going to show you a video of Aisha and Ahmed collecting. Since the volume is low, I'm just going to say what in the slide is. This is Ahmed and Aisha collecting an aromatic plant. This has a very spicy smell to it. And the background of this video is like really windy. Once the plants were collected, they were washed and dried. Here we have Khalida and Aisha. Khalida is just here. Drying the plants in the fume hood. The plants were then separated into 4 parts rather than just aerial and lower as in other studies. Here is Suzanne and Aisha separating the plant parts. This was a very tedious process as many of the plant parts were like quite small. It is really tiny. Here is Aisha again concentrating on removing the leaves. Khalida, Abir and Suzanne are shown here. This is what Suzanne looked like after weeks of separating the plant. Three chemicals were used to extract the plant extracts. There were extracts were then collected onto sterile filter papers, and here we have Ahmed doing the process. Again, here is Hannah and Aisha, living happy as the extraction process was coming to an end. Small discs containing the plant extracts were created using sterilized hole punch, as you can see Suzanne doing here. The discs were then put into labeled containers and placed in zipper bags. Here we have just a few of the antibiotic discs that were made. Aisha will now speak about the last few steps in our process and then some of our best results. And please don't forget the interesting story she's going to tell.
Thank you, Tasneem. Good evening, ladies and gentlemen. My name is Aisha and I'll be talking about the last few steps of our process. After the extraction discs were made, we were then ready for testing using the Kirby-Buer method. This method is used to test antimicrobial properties of the chemicals in the plant. We took swabs of our culture and then spread them evenly over the agar plate, as you can see here with the spread plate. The plant extract discs for each extract were then placed in the middle of the spread plate. These plates were then incubated for about 24 hours to allow the cultures to grow. The zone of inhibition, which is the area around the disc without any microbial growth. A clear zone around the disc was measured. Suzanne and Bakita are here recording the zone of inhibition. The zone of inhibition here was measured for each extract, water, methanol, and hexane for all 4 plant parts, flower, leaf, root, stem, against all the cultures. For our statistical analysis, a 90% confidence level was used on all 19 plants to see which gave the best results. We found 153 statistically significant results for our plants. This is just one example showing the results of Fulicaria undulata. How many plates? 4 plant parts times 3 types of extract times 3 trials per extract into 11 microbes. 396 plates per plant multiplied by 19 desert plants.